Hey guys. So I'm gonna do Prezi notes for genetics because it's not really something I can draw. And then there will be a second video today for how to solve those genetics problems, and I'll go through like a problem or two on the problem set one and problem set two. So you can go watch that. But I first kind of wanted to start with a little just bit about Gregor Mendel because he's considered the father of genetics. Essentially, he was a monk, and he studied sciences. And then I believe his father was like, "You have to choose. Would you rather go to school to become like a lawyer or something he didn't really want to do, or would you rather be a monk?" And so he chose to be a monk. And then when he was in the abbey, this is where he started experimenting with pea plants. And you can actually go to the abbey where he lived, and you can actually see some of strains of pea plants that he actually worked with. Like, obviously, it's not the original pea plants, it'll be descendants of them, but that's really cool. And so, what Mendel was kind of studying was he was studying these concepts of inheritance. And in the 1800s, most people thought like they could see that a child looked like their parents. But what they thought was that everything was blended. So when two people created a child, the child had a mixture or a blend or an intermediate of the parents' traits. So if one partner was really tall and the other partner was very short, the child would be the average height of those two individuals. And like, if you just look at people and you don't necessarily look at their genes, you would assume that that is true because you can see a mixture of traits from both parents. But if that were true, then eventually all variations would be diluted out of a population. If everything is continuously being blended together, we would eventually all look like each other because we would all just be this blend of traits, and we'd all just be this like average height, average skin tone, average hair color, etc. So Mendel used pea plants as they're called model organisms. So in the world of biology, we have a few model organisms. Peas are one of them. A lot of plants. We have these fruit flies called Drosophila melanogaster. There's these C. elegans. Those are some worms, and Mus musculus, which are mice. So we have these like really particular animals that most scientists use when they're testing, when they're doing their scientific research. And the reason is if they're easy to grow and maintain, meaning it's easy to house these things and they don't take a lot of upkeep, and then it's really easy to keep them alive. It's easy to control their mating. So if you want to be testing evolution or genetics or inheritance, you need to be able to control which organism mates with which organism. And so in the case of plants, you can just take the pollen, they use a paintbrush, and they'll just take the pollen from one plant and they'll inseminate the eggs, the ovum from another plant. So they're literally controlling the mating. Same thing with if you're using an animal. So like the mice, you just put them in the same uh, little cage together. So like they're the only ones they can mate with. In the case of the fruit flies, I remember in high school I was in charge of taking care of all the fruit flies. I was a TA for my bio teacher, and you just put them in the same tube, and then a few days later there'll just be a bunch of little baby fruit flies in there. So you can kind of control it. They make lots of babies. So each generation, typically in humans, we create one offspring at a time. But in the case of fruit flies, mice, plants, they're producing many offspring, so their sample size is larger. The traits are really conspicuous, meaning they're really, really easy to determine what trait you have. So if you're looking at pea pod color, it's really easy to see if it's green or yellow. There's no confusion as to what the trait is. And then the generation time is really quick. So you can do your research within a reasonable amount of time, and you can collect a lot of data on multiple generations because their generation time is so quick. So this is what we mean by like conspicuous traits. Like you can really tell if a flower is purple or white, and there's typically two options for the traits instead of multiple. So essentially what Mendel did is he took these plants, so he started with purple and white flowers, and he essentially controlled their mating. And so what he did is on one flower he removed their it's called their stamens, and the stamens are what produce pollen. So if you ever looked at a flower, like the long filamentous thing that has the pollens at the end, that's the stamen. And then inside the carpel, that's where the egg is. So this is essentially like a little uterus here. And so he removed the stamen on one flower so it could not pollinate the other. And so he essentially made like a female flower and a male flower, even though flowers have both parts. And then he just took a paintbrush, he took the pollen from one and he inseminated the other. And then he grew what's called the first filial generation, and this is the first offspring. So when we talk about generations, our first parental line we call the P generation for parent, and then their offspring we call F1, and then if the F1 have offspring, we call them F2, so on and so forth. And so what Mendel found is when he mated the purple flower with the white flower, all of their offspring were purple. And then what he did is he took two of these purple offspring, so like siblings, quote unquote, and he mated them, and he found that he had a 3-to-1 ratio. So he had about 75% purple, 25% white. And he did this over and over and over and over again, and he continued to get this same 3-to-1 ratio. And this puzzled him because first they thought the blending would occur. So he assumed that these flowers would be like a lavender, and they weren't. And then when he mated them again, he was surprised that a white flower popped back up. And so these are all the different traits that he studied. So he started with the flower color, and then he actually moved position. So if flowers are located on the sides and lower on a plant, they're called axial flower buds. And if they're up on the tops of the stems, so they're only found on the ends of the stems, they're called terminal. He looked at the color of the seeds, the shape of the seeds, the shape of the pods, the color of the pods, the shape of the stems. So he studied all these different conspicuous traits of the pea plants, and he typically found an almost 3-to-1 ratio. So it's never exactly 3-to-1 ratio, just due to variability and his sample size. So he never was able to test, like, you can't test every single purple flower and every single white flower in the world, but with his sample size, this was his ratio. So you can kind of see most or all of them are roughly a 3-to-1 ratio. And so these were his two big conclusions. His first conclusion is in organisms that reproduce sexually, so there's sperm and egg, there has to be two different versions of every trait. So a trait could be something like we just looked at. So a trait could be something like seed color. So this is my trait, and these are my two alleles. And so he found out there must be variations of every trait, at least two, and one must be inherited from the mother, and one must be inherited from the father. So he kind of figured out this idea that you get one chromosome. He didn't know about that yet, but like one from your mother, one from your father. The second thing he determined is that there's this idea of dominance and recessive. So this is kind of when the whole blending hypothesis was blown out of the water, because he determined that if you have two different alleles, so if you are what we call heterozygate, where you have a dominant allele and a recessive allele, then the dominant will be expressed over the recessive. And so that was told to him by this F1 generation. So he started figuring out, like, if there are two different alleles, one's dominant and one's recessive, and they're mixed, so heterozygate together, then that's why all these would be purple flowers. And then when he took two heterozygotes, so they're the same phenotype, they have the same color, but they have a different genotype. So this one is what we call homozygous dominant. This one is heterozygous. When he crossed the two heterozygous flowers, then he got that 3-to-1 ratio. Very nice. So just like recap, this stuff you should know. Genotype is the actual genetic makeup. So we either call them homozygous dominant, homozygous recessive, or heterozygous. And then phenotype is what is the actual physical trait being expressed. So think phenotype is the physical characteristic, genotype is the genetic makeup, okay? When we use these capital letters and lowercase letters, those are just symbols. They're just like a widely used symbol system. So if you're ever asked like the genotype of an organism, you wouldn't say big P, big P. You would say they're homozygous dominant, okay? Because these symbols are just like placeholders for these words, homozygous dominant. Okay, so there are two laws that Mendel ended up developing, and the first one is called the law of segregation. And what this law states is that during meiosis, which is the production of gametes, every gamete receives one allele for every trait. So if you remember, every trait we inherited from our parents, and we have two copies of every trait, two alleles. We inherited one from our mother and one from our father. That's why we have 23 pairs of chromosomes, because we got 23 from mom, 23 from dad. When you go through meiosis, you end up with one allele in every gamete. So that's why the cell will start with 46 chromosomes, 23 from mom, 23 from dad, but the gametes, the sperm and the egg, will end with 23 total chromosomes because they're only getting one copy of each allele. It's going to be like a new combination now. So like I have 23 from my mother and 23 from my father. In my eggs, I have only 23, and it's going to be a mixture of mother and father genes. So that's why you and your siblings look slightly different, because the sperm and the egg that were fertilized when they made you had different combinations of alleles from your parents' parents, and then the same thing from your siblings. So you might have inherited like your mother's hair and your father's skin tone, while your sibling may have inherited your father's hair and your mother's skin tone, because things can get recombined when you do meiosis. And so the law of segregation just states the alleles will segregate. This occurs during meiosis, during metaphase and anaphase. Remember, metaphase, all the chromosomes line up on the equator, and then anaphase, they all get pulled apart. So that pulling apart of the chromosomes is pulling apart the alleles of each trait. And you can see that here. So we have all the chromosomes lined up. So, ooh, yes. Okay, so on one side, this side will either be like the mother or the father allele, and then here will be the mother or the father allele. Or sorry, these are all replicates. But like this side might be like chromosome 1, this might be chromosome 1, and then this might be like the father's allele on this side, and this might be the mother's. And how they separate will kind of determine which of the new cells get different alleles from the father versus which of the new cells get different alleles from the mother. As you can see, this one cell has like this chromosome that's mostly blue, and this one is mostly orange, and so it's getting these different combinations of alleles, and that's why all our siblings look slightly different. The second law he came up with is called the law of independent assortment, and this is where alleles for different traits are passed independently of each other. So this is something like hair color and eye color. Just because you have brown or dark hair does not mean you will also have brown or dark eyes. So those are two different traits, two different alleles, and they will assort independently of each other as long as they're found on different chromosomes. So just because you have a hitchhiker's thumb does not mean you also have a widow's peak, because those traits are found on different chromosomes. And so the only time we really see two traits occur together often is when they're what we call linked. And you can kind of see down here, as long as the alleles are unlinked. If they're linked, it means that the genes are found on the same chromosome. So there's a really common example. It's red hair and freckles. So the mutation, the variation of having red hair and the presence of freckles are actually found on the same chromosome. And so because they're found on the same chromosome, The chances of having a crossing over event right here where they exchange genetic material and separating those two genes is really unlikely because red hair, I'm going to zoom in again. Ooh, two zooms in. Red hair might be found like right here on a gene, and then freckles are found right next to it. So the chances of having a crossing over event that would separate red hair and freckles and exchange that material so that you've separated the red hair from the freckles is really, really low. That's why typically people with red hair also have freckles. Could it be that people with red hair don't have freckles? Of course it could happen. It's just a lower chance of happening. And so what Mendel found is that because a pea pod is yellow does not necessarily mean the pea pod will also be wrinkled. And same thing for the green pod. So he looked at two different traits. It's called a two-factor cross. So he crossed yellow round pea seeds and then green wrinkled seeds, and he found that those traits can kind of be recombined. So you can get any combination of those different traits. Ooh, she's meowing. Cool. So recombinant offspring are when you get offspring that are different from what the parent generation looked like. So in this example, he crossed a yellow round and a green wrinkled. And so these offspring that he got, these weren't recombined because they had the same combination of alleles as the parents. But then we got these offspring that did have new combinations because he never had a yellow wrinkled, right? And he never had a green round. And so we call these the recombinant offspring because we recombined the alleles to create new combinations that never existed before. So like you might have new combinations of traits that has never existed before in this universe, and same thing with your siblings. So like you are a completely unique organism. That's crazy. Okay, I'm gonna do that in a separate video next. So that's kind of like what Mendel found. He's pretty much just like straight up dominance and recessive. But since Mendel, we've kind of discovered that there are other forms of inheritance that don't follow the dominance and recessive rule. Doesn't mean that he was wrong. It just means that we've kind of like built upon everything he found out. So we call these non-Mendelian genetics or extensions to Mendelian. And so we're going to kind of go through 1, 2, 3, 4 different types of like non-Mendelian, 4, 5, 6 different types. So you should kind of like know each type in general. Um, so like Mendel figured out like simple dominance, like the dominant gene will always be expressed over the recessive gene. And what we found is there are certain organisms that have certain genes that don't display that. They do show the blending that was thought in the 1800s. And so for incomplete dominance, when you get a heterozygote, I really like these flowers because it looks like this one is singing. Like, look at its hair and its cute little arms. Oh, so cute. Sorry. So in incomplete dominance, neither allele is completely dominant. So because neither allele is completely dominant, when the genes are expressed, they create proteins. And those proteins, which are both produced, so we're producing the white and the red protein, they are produced in equal amounts, and you end up with this blended look. And so the proteins are essentially blending together to create this intermediate color. So when you have the heterozygote, it has one of each allele, you end up creating an entirely new phenotype. So if you remember from simple dominance, there's only two phenotypes. There's the dominant and there's the recessive. In incomplete dominant, neither of them are dominant or recessive, so we can't even label them that. We just call them white and red. And when you have the heterozygote, you have a blend of the two alleles. So it alters ratios, right? Because instead of having like that 3-to-1 ratio we would expect, we have a 1-to-2-to-1 ratio. So we have 1 red, 2 pink, 1 white. We also have this phenomenon called codominance. So with codominance, it's instead of having this incomplete dominance where neither is dominant, they're both dominant. So both alleles are equally dominant. And because they're equally dominant, they're both expressed in equal amounts. And so our most prime example of this is blood typing. So if you're somebody who has A blood, type A, it means on your red blood cells you produce this carbohydrate, which is, we call it the A antigen. And then if you have type B, you produce the B antigen. If you're type O, you don't have any. So people with type O blood, you actually don't have any of these carbohydrates and proteins on your red blood cells. If you have type A, you have the A antigens. If you have B, you have the B antigens. If you have AB, this is our example of codominance, you get both. And so they're both equally dominant. So you see both of the alleles in equal amounts, which is really cool. The next type is when you have traits that are controlled by more than two alleles. Like so far, we've been talking about there's only really like two alleles for everything. There's like a pink or a red. There's a purple or a white. But many of our traits are actually controlled by many alleles. So multiple alleles, this is the blood typing. So instead of just having like a dominant or recessive, we actually have three different alleles, and they're right here. So we have IA, this is a dominant A allele. It's equally dominant, so it's going to be equals. It's equally dominant to B. And then both of those are dominant, so greater than over I. So the recessive I allele is representing our O blood type. So our genotypes would be right here. Well, this is fun. So these are my three different alleles. So if your father had type O blood, he must have given you a recessive allele because that's all he can give you because he must have been this genotype. If your mother was type B blood, she has a couple options, right? Because we could either be homozygous dominant for B, or we could be heterozygous because O is recessive. So if two people, a type O and a type B blood person, have an offspring, they can either have offspring with type O blood, or they can have offspring with type B blood. More likely their offspring will be type B blood if they are homozygous. So like if they're homozygous dominant, it has to be B blood. Okay, so this one's controlled by three different alleles instead of just two. And so what happens is the ratios get insane because you end up having, instead of just like two different phenotypes, you can have four different phenotypes show up in all these different blood types. And then genotype wise, you can have six different genotypes instead of three. So the more alleles that are involved with a trait, the more variation you can have within the genotype and phenotype. And so this brings us to polygenic inheritance, when more than one gene contributes to the expression. So in multiple alleles, it'll be one gene, but there's just three different variations of it. So instead of just having like a purple flower and a white flower, there would be a third option. And so then I would get any combination, any two of those would give me my blood type. So with skin color, height, hair color, these are polygenic genes. So polygenic means instead of just one gene controlling this trait, it's multiple genes controlling this trait. So for human skin color, when you have this spectrum of phenotype from the pale to the darker, it means that there has to be multiple genes involved because we have so many different phenotypes. There has to be that many alleles involved. And so in the case of skin color, height, hair color, the shade of your skin or hair or the height that you are is determined by how many dominant alleles you inherit. So if you take a heterozygous individual crossed with a heterozygous individual, you could get an individual, like an offspring, that is completely recessive. So recessive would be our palest skin tone, and then we have variations of that. So as you add a dominant allele, they become slightly darker. So you can see all these with the one dominant allele are slightly darker, and you can get two dominant alleles, they're slightly darker, three dominant alleles, slightly darker, until you get completely dominant allele, which would be our darkest skin tone. So the same thing would apply for height. If you take two people that are average height, they could have an offspring that is very, very short or an offspring that is very, very tall. right? So if two tall individuals mate, they're probably somewhere down here in the genotype. It's more likely that those tall people will also have a tall offspring because they have more dominant alleles to pass on. People who are shorter will most likely have an offspring who is also shorter because they have more recessive traits, alleles to pass on. But does it necessarily mean you're going to be as short or as tall as them? No, due to the law of segregation and independent assortment. Like the recombination could be anything. So cool. Okay, and now we have these interactions between genes. So those were kind of like all our different types of inheritance. And now we can have genes like that interact with each other. And so like multiple traits are being affected. So pleiotropy is when one gene has multiple effects on the physiology of an organism. So our prime example is sickle cell anemia. Sickle cell anemia is caused by a point mutation. It's caused by a single substitution. It's a missense mutation. And that single point mutation causes the shape of the blood cells to change from this donut shape with a little indent to a sickle shape. Okay. And what happens because of that is now we've changed the phenotype of our blood cells, but this also has an impact on the physiology of the organism because now these sickle-shaped blood cells are more likely to get caught on each other and also get caught on little grooves or protuberances in your blood vessels. And so people who have sickle cell anemia tend to have blood clots occur really easily because their blood cells are catching on each other. And then that blood clot can lead to cardiac arrest, and you have all these other physiological effects due to one single change in one gene. Epistasis is when two or more genes control the expression of a single trait. So in polygenic, you had many genes kind of combining to give you the variance of a trait. And with epistasis, you have two or more genes, and they're all kind of responsible for a different aspect of the trait. So, like with skin tone, they were all expressing the same thing, the darkness of the skin. With epistasis, the genes are not expressing the same thing. Let me explain this better. So in the case of Labradors, if you have a Labrador, there's only three options for Labradors, right? Like you could have a black lab, a chocolate lab, or you could have a yellow lab. And so there's actually two genes that control Labrador coat color. And the first gene is called the E allele. And what it does is it just produces the chances of a Labrador producing pigment or not. So we have one gene that's just like pigment production. You have a second gene that is color of the pigment. So these two genes together can give us the color of our Labradors. If it was just this one gene, color of coat, we would only have two colored Labradors. We wouldn't have yellow labs. But because we have an epistatic trait, we have to look at, is there pigment, yes or no? If there is no pigment, meaning you're homozygous recessive, that means you have technically an albino fur colored Labrador. This is just for the fur. So like they're not albino skin tone, they're just albino fur color. So they have no pigment in their fur. So they appear yellow when light bounces off those pigments. Or sorry, lack of pigments, when they bounce off the keratin in the hair. So presence of pigment, yes or no? If no, yellow lab. If yes, so if you either have a heterozygous or homozygous dominant, then now we have to look at, so then what color is the fur? And then we look at our B allele. And then chocolate is recessive. So if you have homozygous recessive, you'll have these chocolate labs. If you have dominant, homozygous dominant, or heterozygous, you'll have the black labs. So now we have two different genes controlling the same trait. So they're not controlling the same aspect of the trait, but they're controlling the same trait. Skip that. This is just a sum up. Yep. This is kind of talking about the linkage I was telling you. So if genes are on the same chromosomes, remember we have chromosomes 1 through 22, and then we have our sex chromosomes, we call them linked. So if two genes are found on the same chromosome, like so like this B and the VG gene are found on the same chromosome, the chances of there being a crossing over event to separate them is lower if they were compared to if they were found on different genes, different chromosomes. So genes that are linked are more likely to occur and travel together. So they're more likely to show up in the offspring together than genes that are separate. And then we have this thing called sex linkage. And these are genes that are found on our sex chromosomes. So typically we look at the X chromosome when we look at sex-linked traits because it carries more genes. And the examples that we use are typically things like color blindness, male pattern baldness, and stuff like that. So if you think about anybody you know who's colorblind or anyone you know who's displaying pattern baldness, those individuals are typically male. And so males actually show sex-linked traits at a higher frequency than females. Is it possible for a female to have pattern baldness and colorblindness? Yes. Is it likely? No. And the reason is, for a woman to have colorblindness, if it's carried on the X chromosome, so the colorblind allele is here, for me to inherit colorblindness, I would have to inherited it from my mother and from my father because I have an X and an X chromosome. So the likelihood of them both carrying the colorblind allele is really low. For a male to be colorblind, because his genotype is XY, here's the Y chromosome, he would only need to receive an X chromosome from his mother that has the colorblind allele, and he would express colorblindness. So the chances of a male inheriting one allele versus a female inheriting two alleles is just higher. And so if you are colorblind, or if you are experiencing male pattern baldness, it's actually due to the X chromosome you inherited from your mother, but she inherited that X chromosome from her father or her mother. So if you're wondering, like, am I going to go bald if you're a male? If you look at your mother's father, so your maternal grandfather, if he is balding or has male pattern baldness, it's possible that your mother inherited his X chromosome, and then you might also go bald. I'm sorry, that's a deep cutting revelation right there. Okay, we're gonna skip over all of this. I actually think we might be done. Oh yeah, last thing I wanted to talk about. So we've kind of just talked about, like, if you are homozygous dominant, homozygous recessive, heterozygous, like, this is what you will look like. There are actually other factors that can control your phenotype and your gene expression. So if you've ever seen the flower hydrangeas, mostly they occur like this purple color, but if you actually change the pH of their soil, so if you make it more acidic or more basic, you can actually alter the color of your hydrangeas. And the reason is that I don't know if it's the hydrogens or the hydroxides, but some factor in the soil actually interacts with the genes, and it causes them to turn on and turn off. And so we're only expressing one of the alleles or both of the alleles, and it's causing the color of the hydrangeas to change. There are certain animals that actually change their coat color on a seasonal basis, and this would be due to temperature. So as the temperature changes in their environment, it actually causes certain genes to be turned on and turned off, and then it causes their phenotypes to change. So their genotypes are the same. We're just choosing, like, which allele is expressed instead of both at which time. And so, like, this was a mutation, right? Like, there was a rabbit that was that was produced, that had this mutation where in cooler temperatures it became white, and in warmer temperatures it was brown. But think of natural selection, right? Like, if this rabbit lives in an area where it snows in the winter and it does not in the summer, then that would provide an advantage for this rabbit. And so, like, that variation where the environment influences their coat colors persisted throughout the population because it increased their survival. So cool. All right. That's it for genetics. I'm going to put the links to these prezzies on the document so you can take notes if I went too fast. And then go watch the next video on how to solve genetics problems. All right. Bye, guys.